introductory talk to the symposium we're putting on, Ecology and the Evolution of Reproductive Traits. We're really excited to present this symposium to you all and to have put it together. So we'll kind of start off with thinking about the relationship between ecology and evolution. So really these are driven by the same underlying processes, right? And I think we all recognize this, that births and deaths are ultimately what's going to change population size, and these are also the proximate cause of any change in allele frequencies. So despite the fact, first of all, this kind of seems obvious, right? But also, I think we maybe don't recognize just how deep this connection can be. There has been a really long and rich history of thinking about these things. So, at least going back to Hutchinson, the ecological theater and the evolutionary play, Lewontin talked about these topics as well, and more recently, kind of with the understanding that evolution can occur on contemporary timescales. This has been showing up in the context of eco-evolutionary dynamics as well, right? So really an active area of research, but also has this really long history. Despite this fact, typically in evolutionary biology, when we think about the role of ecology, we're thinking about traits that directly modify species interactions. So a couple of examples of this. One would be maybe the evolution of some interaction rate directly, for example, this paper on the evolution of predation rate. Also, there's been a lot of work in the context of competition with this, for example, character displacement and dynamics similar to that. So the question then becomes, and what's kind of motivating this symposium is, what about traits that don't have this really obvious direct ecological relevance? Is there still an important, what's the role of ecology in that context? And specifically for this symposium today, we're going to be talking about reproductive traits. And I think there's really a lot of good reasons to think that ecology would be really important in this context. So first of all, the benefits of reproductive decisions can depend on ecological context. So this is an example in spadefoot toads where hybridization and potentially mate choice based on hybrids is going to depend on how dry the year was. It's also true that reproductive decisions can affect population viability. So there's a lot of work on sexual selection and extinction risk. This example is from ostracods, which showed that sexual size dimorphism predicted extinction risk. Reproductive decisions can also directly influence population dynamics. This is from a model of divorce showing that divorce decisions can affect population density. And finally, reproductive decisions can be directly influenced by ecological factors. So again, there's kind of a lot of work showing predation influencing mate choice. This is from an example from guppies showing that females are less choosy in high predation environments. So there's all of these reasons to think that ecology might be important for reproductive traits. In my own work, kind of connected to that last guppy example, I've thought a lot about the potential of predation to influence sexual selection. So if you have this model where you mechanistically treat the cost to expressing a sexual display as increasing your predation risk, then in examples where you have predator-prey cycles, this gives you fluctuating selection. And as a result of that, you can get cycles in the frequency or the, extravagance of a sexual display. And kind of as a result of this project, it's something that I was thinking a lot about, and it was really evolution, I guess, a year ago that it really hit me just how much a lot of people were thinking about these topics. So this is just kind of a set of papers that were published in the last couple of years showing a lot of diverse ways that people are thinking about the role of ecology in reproductive decisions or reproductive traits. So this first example, connecting thermal ecology with sexual selection. Second example there is a species of cricket that's changing its sexual signal as a result of parasitism pressure, a model on how ecology can influence the evolution of sex roles, Even in the context of community ecology, people are starting to think about the importance of sexual interactions. And then finally, the last example showing that climate change might be affecting divorce decisions in albatrosses. So there's really this wide range of examples and wide range of reasons that people are ways in which people are studying how ecology can influence reproductive traits. And it really kind of came together for me, like I said, at Evolution last year, where it's like, wow, there's a lot of work being done on this. But it seems like, I've never seen, there's not very many review papers, for instance, or it didn't feel to me like its own kind of unique subfield. And that's kind of what motivated us to want to put this symposium together, was to bring together, all of these diverse ways that people are studying how ecology can influence reproductive traits, and to kind of, point out that this is something that a lot of people are studying, and that it is sort of its own, I guess, subfield. So I'm going to turn it over to Sarah, who's going to talk more about the symposium.
Yeah, thanks. So Brian's done a really nice introduction to the sort of theory behind why this is a topic that we thought was relevant. But why did the two of us in particular propose this symposium? I was a postdoctoral fellow at the National Institute for Mathematical and Biological Synthesis at the University of Tennessee, Knoxville. And I think it was in 2017 that Brian was an undergraduate researcher who worked with me and a team of three other undergraduates, putting together a mathematical model where we were trying to predict how migration timing and sex differences between competition and migration timing might impact reproductive outcomes in a made-up population of birds. So this is a picture of Brian and his three teammates, all pretending to be the birds that they were modeling. And so it was a pretty fun model to put together. Brian worked really hard to help make sure it got published afterwards. And so we've both been interested together in the impacts of ecology on reproductive decisions. And one of the things about this model that I thought could be a really potential next step that we are not following up on at all because we've now moved on to other things, is putting this model into a more evolutionary context. So we only modeled a single breeding season, essentially, but how would this impact on evolutionary timescales? One of the things the reviewers wanted to know, and we thought was beyond the scope of our undergraduate research project. And so I reconnected with Brian at the Evolution meeting last year, and we basically kept running into each other as we were running from one session to another to try to go to the talks that we thought were really interesting. And so we realized that this is a set of topics that were dispersed across many different sessions at last year's Evolution meeting. So we thought, hey, why not propose a symposium so that I don't have to run around the convention center quite as much? And so we think that, or the response to our symposium is indicative that our topic is indeed highly relevant. We received over 100 abstracts, of which more than half ranked us as the first choice, which was really a positive as well as daunting outcome of proposing the symposium. So in case you're curious, Brian and I ranked each of those 102 abstracts independently. We retained all of the talks that we both ranked in the top 20 and then filled in the remaining slots going one by one down our ranked list, and ended up with 19 really exciting talks. And our approach resulted in speakers from 8 different countries spanning 3 continents. I did not put my or Brian's locations on here because it wouldn't have changed the countries, but we have speakers from North America, Europe, and New Zealand, which is excellent. And so we have this really diverse range of people. And based on a little bit of light internet stalking, I believe our speakers range career stages from undergraduate researcher through to full professor. And the talks in this symposium cover a really broad range of organisms, Hopefully I found correct Creative Commons pictures for your organism of choice if you're one of the speakers in this symposium. But we have animals and plants and ranging from, corals and parasites to fish and birds and all sorts of plants and pollinators. And so we're going to have a really diverse set of talks during this symposium. I've also put up this, little matrix picture to represent our theoretical talks that we have, because it turns out when you look for an image of evolutionary modeling, nothing really dynamic comes up, but the matrix sort of picture seemed to cover that. As a broad overview of the types of things we'll be hearing about throughout this symposium, this is a word cloud of all of the abstracts. So there's very clearly a lot of talk about reproductive traits, evolution, selection, offspring, species, mating, behaviors, populations, evolutionary. This is not a surprising word cloud, but it is an indication of some of the common themes that are popping up throughout the symposium. So once we had our selected abstracts, we allowed the sort of properties of those abstracts to emerge into 4 research themes, very broadly speaking, where in this session we'll be hearing about plant reproduction and coevolution with pollinators. Our second session will be covering sort of mechanistic approaches towards understanding reproductive trait evolution, including various omics techniques. We'll have a session on how environmental influences can impact reproductive traits. And our final session is roughly sexual selection and reproductive behaviors in an ecological context. I was really excited when I saw these themes start to emerge because 3 of these themes are directly related to the research that I do in my research group, where I use a comparative approach integrating behavior, ecology, and genomics and transcriptomics to try to understand the evolution of ornaments in a group of marine fish called pipefish. If you want to learn a little bit about some very preliminary data that I have, please come to my poster in poster session 2. And you can see these pictures that my students have drawn of these beautiful pipefish in a closer look. But these 4 categories also integrate the various topics that Brian introduced to us. So thinking about species interactions impacting evolutionary trajectories, influences of ecological factors, reproductive decisions impacting on population dynamics and viability, and the benefits of decisions are dependent on ecological context. So I believe that all 4 of these themes will come throughout the entire symposium, and that we'll be seeing at least 3 of those 4 themes in each of our different sessions. So I am very excited to hear from all of our speakers today, and I hope that many of you will come along to the rest of to all of our sessions. So this is just our wonderful evolution system, highlighting that we have this session now, a coffee break, our session again, lunch break, another session, coffee, and then our session again. So please do stay with us, and I look forward to hearing from all of our speakers today. And we've got a minute to turn over, so perfect.
Okay. Good morning. So, bienvenue à Montréal. So I'd like to start by thanking Brian and Sarah for inviting me to present to you this morning. So, and I'll be presenting some work we've been doing in the lab in the recent years. So we know that plants, flowers, are importantly shaped by their pollinators. And over the years, there's been lots of attention given to specialization or adaptation to their specific pollinators. And we know that because of expression like adaptation to the most effective pollinators that was coined by Stebbins in 1971, or just concept like pollination syndromes. However, it's increasingly known right now that specialization is more the exception than the rule. Here you have two examples of community surveys, and on the x-axis you have the number of visitor species to different flowers. And as you can see, there's a lot more species that attract a lot of pollinators than species that attract very few pollinators. And so generalism in pollination is probably much more frequent than we previously thought. And this has important implications. For instance, it's been suggested that generalists result normally in greater intraspecific variation. There's also impact on selection. For example, there could be complex interactions in fitness function and trade-off that affects selection, and also extinction, where it's been proposed that generalist strategies result in reduced extinction rates. So these are the type of questions we've been trying to answer in our model group in the lab, which is the Antienne-Gessneriaceae. So this is a group of about 80 species that is monophyletic and strictly found in the Antilles in Central America. It's a group that is highly diverse in both shape and colors, and this variation is tightly linked with their pollinators. So there's three main pollination syndrome in that group. Two of those are specialists. So there's a hummingbird syndrome, which is associated with bright tubular corollas and diurnal production of nectar. There's also a bat syndrome with a campanulate corolla with pale colors and a nocturnal production of nectar. And finally, there's also a generalist syndrome. Surprisingly, this has converged to a very specific flower shape, which we call a subcampanulate corolla, because it has a constriction at the base of the corolla, which is very important for the fit of the pollinator with the corolla. And those have both diurnal and nocturnal antitheses. So before we go forward, I thought I would define what I mean by a pollination generalist. So in the group, we define generalism as a species that can be pollinated by a different functional group. So in this specific group we're studying, the generalists are pollinated by hummingbird, bats, but also insects. And if a species is pollinated by several species of hummingbirds, then that's considered to be a specialist. What's interesting in this group is there's been several transitions between those three major pollination syndromes. So there's been at least three independent origins between each pair of syndromes. And these evolutionary replication makes it a very good group to study evolution of flower morphology. So in this presentation, I'm going to talk to you about two specific studies we've done recently. So one is looking at the impact of generalist pollination strategies on intraspecific variation in the group, and the other on the evolutionary correlations between the shape of flowers and that of the bill of their hummingbird pollinators. So let me first talk about some of the methods we've been using for those studies. The first one is for flower shape characterization. So we've been using geometric morphometrics. That involves landmark positioning. You can see here on your left that the blue points are the landmarks. So these are homologous position among the different flowers, and the red dots are semi-landmarks, and they are used to capture the overall shape of the corolla. And once we have determined those points for each flower, we use general Procrustes analyses that removes translation, rotation, and size to be able to really compare the shape of the different flowers with each other. We also base our results on lots of pollination data that was almost all collected by Silvana Martin Rodriguez. So we have data, very good pollinator data on 29 plant species, and that is very important for the studies we've been doing. So let's start by looking at the impact of generalist strategies on intraspecific floral variation, and this was work that was led by Marion Leménager for her PhD student. Marion just recently defended her thesis from the lab, and that's work done in collaboration with Abel and Marales Castro, Silvana Martin Rodriguez, and John Clark. And that paper is presently in press in Journal of Evolutionary Biology. So the motivation behind this study is that this hypothesis coined by Van Vellen in 1966, who proposed that generalists should have greater intraspecific variation than specialists. And this is because the generalists had to fill a greater ecological niche. This hypothesis have been tested a few times through time with some mixed success. In some cases, it was confirmed that there was greater variation in generalists, others not. But there were very few examples of tests that involve a whole group of organisms. And this is really what we wanted to do here using evolutionary approaches. So we tested this hypothesis that generalists have greater variance than specialists with 3 different methods. The first one is a phylogenetic generalized least square, and this is what I'm going to be presenting to you today. We also use evolutionary methods to test that, but I won't have time to talk about those. I encourage you to go and have a look at the manuscript that is online. So this is what the data looks like. You have the phylogeny on the left with the different flower shapes, and you have a representation of the morphological variance in a log scale for each of the species. The specialists are in red and the generalists are in green. And on the right here, you have the species name with the sample size in parentheses. So you can see that in general, the generalists seem to have a greater variation than specialists. And that was confirmed with the phylogenetic generalized least square, where the generalist syndromes was highly significant. So it does seem that the niche variation hypothesis is confirmed with that group, and so that the generalist species have greater intraspecific morphological variation than specialists. So why is that the case? Where there are 2 potential scenarios that might explain greater variation in generalists. One is if the generalists consist of individuals that are specialists, like several specialist species that are specialized for a different part of the ecological niche, like the top right picture here. And the other alternative is if the generalists consist of individuals that are themselves generalists. And it seems like this second option is the case here in that group, because the flowers of those generalist species can all be pollinated by the different groups of pollinators. So the greater variation then might be just due to relaxed selection, or maybe adaptation to different proportion of the different pollinators in different parts of the range of those species. So now for the second part, I'm going to talk to you about the impact of general strategies on the evolutionary correlation between flowers and pollinator shape. And this is a project that was led by Joly, who was also a PhD in the lab, and that was done with the collaboration of Silvana Martin Rodriguez and John Clark. So the motivation behind this study is that there's been several studies that have looked at the relationship between the bill length or curvature of hummingbirds and the corolla length of curvature of the flowers. And there's been several times a positive relationship has been found, but those studies have only looked at hummingbird specialist species. They haven't looked at species that were generalists in pollination, and most studies have used strict linear measurements to look at this correlation. So we wanted to test this relationship in this group to see whether we can find such a positive relationship, and whether the relationship was the same for specialists and generalists. So in other words, if the ecological strategy has an impact on those evolutionary correlations. So we characterize shape using a combination of both univariate traits, so both length and curvature for the bills of hummingbirds and the flowers, but also using multivariate shape using geometric morphometrics. We also use pollinator information for 18 different plant species and 6 hummingbird species. And here you have a few examples of a pair of a Gesneriad species and their hummingbird pollinator. So I'm going to first show you the results for the univariate analysis, which was done using phylogenetic generalized least square. So we tested how bill length and curvature could predict corolla length and curvature. And we tested the predictor, the pollination strategy, and their interaction. If the interaction was significant, that means that the specialists and generalists have different evolutionary correlation for the link between the bill and the corolla. So on this, the results are on the right. On the first row, you have the regression of corolla length, and on the second row, you have regression of corolla curvature, and on the left column, you have the effect of bill length, and on the right of bill curvature. So for corolla length on the first row, you can see that we found a positive significant relationship for bill length. There was no significant interaction, but that might be due to a small sample size, because you can see that the points in blue here, the relationship does not seem to be very strong for the generalist. However, when we look at corolla curvature, there were significant interaction for both bill length and bill curvature. That suggests a positive relationship for the specialist and a negative relationship for the generalist. So that suggests that the ecological strategies of species do have an impact on the evolutionary correlations between the bill shape and the shape of the hummingbird, the plant species they pollinate. That was confirmed with the approach we use with the geometric morphometrics. So we tested that using phylogenetic two-block partial least square. So you can see here that the correlation for specialists is significant. You can see here this correlation on the bottom left. You can see that curved bills are associated with curved corolla, and on the top right of the graph, you have straight bills that are associated with straight corollas. And the generalists didn't show any significant correlation between the bills and the flower shape. So that also suggests that the ecological strategies of the species do have an impact on the evolutionary correlation of those traits. So in conclusion, I hope I convinced you that pollination generalist strategies did result in an increase in intraspecific morphological variation in this group of plants, and also that it reduces the evolutionary correlation between the shape of the flowers and that of the bill of their hummingbird pollinators. And with that, I would like to thank you for listening to this talk. Yep.
How much of a cost is there to specialization for both the pollinator and the plant?
It's a good question, and we don't really know. And we've tested different things. We thought there was a cost because those are species that are found in islands. And there's much more fluctuation in the pollinator communities on islands. So we thought there might be a cost for specialization in these islands, and that might explain why there was evolution towards generalism. But interestingly, there are still a lot of species that are specialists and that are evolving towards specialist strategies, so that cost doesn't seem to be like super important. Yeah. Okay. You can come and see me afterwards if you want.
Good morning, everyone. My name is Autin Cruz. I'm a PhD candidate at the University of Arizona, and I'm really excited to talk to you about some of my research titled The Phylogenetic Co-occurrence of Pollination and Dispersal Mutualisms in Plants. So to get things started, the study of how species interactions affect macroevolution and macroevolutionary patterns and processes, which in turn shape and can shape species interactions, is a growing and important field in evolutionary biology. One really great model in which to study this is in plant systems. And here I'm showing you this legume system with its 4 different interactions, 4 different potential mutualistic interactions, starting with pollination at the top here, with kind of bees and birds. Next, seed dispersal, kind of indicated by these bright red arrows attached to these seeds. Next, we have interaction with microbes via the root system and the exchange of resources. And then finally, we have the provision of defensive services by insects in exchange for extrafloral nectar and a place to live, in this case with these ants. Now, stepping back a bit, we know that plant-associated mutualisms are phylogenetically widespread and well-characterized, and we see this across these mutualisms that I just mentioned in pollination, seed dispersal, defense, and resource exchange. And these are all really great studies published over the past decade or so that get into this. However, the co-occurrence among these mutualisms are really less studied and less understood. There's some work going back about 30 years or so in pollination and seed dispersal mutualisms, and then more recent work in defense and shelter mutualisms that looks at, again, these co-occurrence among them and the evolutionary kind of evolutionary rates as well. Now, I think this co-occurrence is important because, at least going back to our toy system here, specifically with pollination and seed dispersal, because there's a direct correlation and direct impact on plant fitness. We know that most angiosperms, 88% of angiosperms, are biotically pollinated for over 86% of angiosperm history, and there's biotic seed dispersal for approximately 56% of angiosperms. This was an estimate done probably, over a decade ago, but we really aren't as certain with what kind of mode is most dominant within seed dispersal. And I think, this honing in on these two interactions can kind of test at some really interesting, or we can test some really interesting questions here. And so with this schema, with biotic pollination or otherwise, we can look at pollination and seed dispersal kind of in this fashion. So abiotic pollination, abiotic seed dispersal. Here at the top, we have seed-bearing, ovule-bearing cones and pollen-bearing cones with wind dispersal in this first row species. Next, we have abiotic pollination characterized by really reduced flowers, inconspicuous, often non-rewarding with rewarding fruit, so biotic seed dispersal. Next, we have biotic pollination with really showy flowers, rewarding flowers, often specialized, coupled with abiotic seed dispersal, in this case, dehiscent fruit, wind scattered, gravity scattered. And then finally, biotic pollination, showy flowers, fragrant flowers, rewarding, enticing to pollinators with biotic seed dispersal, in this case, rewarding to frugivores in the form of berries. And so kind of with this schema, I want to go into thinking about looking at larger scale patterns of mutualism co-occurrence. And so also, pardon the PDF non-cascading, so bear with me, thank you. So to start off with my first question I'm interested in is, are plants that have pollination mutualism likely to also have seed dispersal mutualism? And to kind of get more specific, does the rate of evolving a seed dispersal mutualism depend on if the plant already has a pollination mutualism? I've listed several different hypotheses as for that second question, the first being that they're independent of each other. There's no kind of coordinated evolution. The second, that seed dispersal mutualism is dependent upon pollination mutualism, but not the reverse. Pollination mutualism is dependent upon seed dispersal mutualism, but not the reverse for the third. And then finally, that they're interdependent of each other, so that they're kind of mutually evolving. So to go about answering this question, I went to the tri-plant database and requested all open access pollination and dispersal traits for anything that was available, did all my cleaning, curation, et cetera, in R, and then I coded them in binary states, abiotic, biotic for both traits, ended up with over 9,300 species, and then built my phylogenetic backbone in PhyloMaker, and here's some information on the tip arrangement and some assumptions about the tree. So really, to look at the evolutionary correlation of discrete traits, we can think about two models, as I was kind of getting at. First, the independent model, which is here on my left, where we have each trait evolving independently in some forward and backward state between abiotic and biotic at some transition rate Q in a Markov process. So for both pollination and dispersal, moving independently. And then in our dependent model, we have both traits correlated, and they're moving together between these four states. And you'll notice that there's 8 transition rates on the dependent model, 4 in the independent model, and we're going to estimate those. So to do that, we're looking at Pagel's test of correlated evolution. This is a maximum likelihood method with k-states, 2 states. I assumed all rates were different in this model. I'm going to test the likelihood of each of these models with likelihood ratio tests, and then I'm going to select amongst the hypotheses or models with AIC, and I'm going to do this in PhiTools. Thanks, Liam. Results. So to kind of real big picture here, there was really large family diversity represented in these data, which is kind of what just came, right? 196 families characterized by some of the big names that we know: Asteraceae, first, Fabaceae, Orchidaceae, Melastomataceae, et cetera. I won't show you here where these data came from, but come see me after. We can talk about where these data come from geographically, and I think we can talk about that after. Next, I found that biotic pollination and abiotic seed dispersal were the most common co-occurrence. And this is kind of counter to what was previously estimated, where there was both dominant biotic pollination and majority biotically seed dispersed by animals. In this case, we found biotic pollination, abiotic seed dispersal. 68% of the data were come from, were categorized like that. And then the next were biotic pollination and biotic seed dispersal, 26% of those data. Here I'm showing you the phylogenetic distribution of mutualism co-occurrence. So these two traits, pollination mode and seed dispersal mode, starting with the inner ring, this blue ring here, blue and white, is the blue being the biotic pollination, and unsurprising, most of these plants are biotically pollinated, with some whites there really categorized by or characterized by Poaceae. Second ring being dispersal mode. You'll notice kind of a smattering of pink and purple, and kind of if you squint, you can see some general patterns. Most eudicots are going to have kind of intermixed with dispersal modes, and then finally at the outer label, or at the outer ring, I've labeled kind of big taxonomic groups just for visual reference. And then finally, I found that seed dispersal mode was dependent upon pollination mode. So the dependent model was statistically more likely with these data, and I'm showing you the transition rates between each state for each model. And you'll notice for the dependent model, which I think was interesting, once pollination gained once pollination gained biotic mode and dispersal gained also biotic mode, that the transition back was predicted to be 0. So there was no transition backwards. They didn't lose that once it was gained. So kind of to recap, are plants that have pollination mutualism likely to also have seed dispersal mutualism? I found no. Biotic pollination, abiotic seed dispersal were the most common co-occurrence. Does the rate of evolving seed dispersal mutualism depend on if the plant already has pollination mutualism? Yes, and we found that seed dispersal mutualism was dependent upon pollination mutualism indeed. So quickly, I want to get to some questions. Where do we go from here? Please don't read this quickly, don't squint. I'm sorry for the PDF version. But this was a really proof of concept. I came in with this kind of curiosity about why we don't really know yet precisely why seed dispersal, or what was the most common kind of mode. Again, mind you, this is kind of coarse grain, abiotic, biotic bins, but we really don't know and don't have a precise estimate of the most dominant seed dispersal mode. I found that in this case it's abiotic dispersal coupled with biotic pollination. I have some really, I think about mechanism. I go to mechanism, and I would love to hear kind of some of your thoughts about some of these mechanisms driving some of these patterns. I won't get into that here. But there's some really great work, theory and otherwise, looking at how autocorrelation in pollination can kind of select for different dispersal modes, both in the tropics and otherwise. And we can also think about kind of hypotheses about physiological constraints as well, and resource-based constraints, with shuttling resources into reproduction, how those might be correlated and coupled together. I didn't control for phylogenetic kind of sampling here. There's some methods to do that. And I think my next steps for this work is looking at multi-state. So looking at, biotic, abiotic, and mixed. There's mixed syndromes as well. and accounting for that with multiple mutualisms. So kind of having taking a Bayesian approach to this. And then finally, just outright, there's some work looking at evolutionary correlations among these multiple mutualisms in trees. This was out in New Phytologist looking at dispersal, pollination, and resource exchange. So that's really interesting work, and I think certainly complementary. So with that, I want to say thank you for coming to the talk. I want to thank my collaborators. I think I've seen them in the audience. Thanks to the Symposium Congress organizers and a few friends that have given me feedback. So with that, almost at 12 minutes, I will take your questions. Thank you. Yeah.
So for the pollinators, there was no back once the mutualism, there wasn't a back. Does that also true for, or kind of true for the seed dispersal? I missed that.
That was, yeah. So both pollination and seed dispersal, there was no back transition, at least with these data. So yeah. Question?
So pollination isn't always a mutualism. Of course, there are deceptive species. Were you able to account for that? Was that data that you had access to?
No, but that's a great point. Yeah, the data that I requested, or the data that I curated, didn't have that fine of detail. But I think there's certainly I could build models into accounting for kind of deceptive pollination and how that might, further affect the correlations.
Thanks.
Yeah, cool question. Yeah, no, that's a great question. Thanks, Priyanka. I think about that. That's what keeps me up at night is physiologically, how can this be explained? And my hunch was that there's these kind of physiological, energetic demands of showy flowers or flowers that are going to kind of take away resources from, producing fleshy, bright, attractive fruits. And that's kind of maybe supported with these data. I think that's part of the whole next step of this project. So I think for all, maybe evolutionary physiologists out there, I would love to pick your brain. But that's really kind of where my head is with this with this work, and I would love to explore more. But yeah, thanks for raising that question. Cool.
So hello everyone, my name is Elisabeth Authier. I'm a PhD candidate at the University of Zurich, and today I will present some of the results of my PhD project. So plant pollination interaction are very important for both pollinators and for many of flowering plants. However, we have seen in the last few decades a decline of pollinators. This decline is both a decline of the overall number of pollinators, but also a decrease of the diversity inside the pollinator community. And because of this disturbance of the pollinator community, we can also expect a negative impact on the reproductive success of plants. During my PhD project, I wanted to investigate this potential adaptive response of plants. And thanks to some studies, we know that there is maybe two potential answers. So the first one, it's a change maybe of the attractiveness. So for example, by increasing attractiveness to pollinators or by discarding some unused attractive traits. Another response could be a change of the ability to reproduce by selfing. So for example, by reducing the barriers to the molecular, physical, and genetic barriers to inbreeding. Thanks to recent experimental evolution, we know that both attractiveness and the selfing ability can evolve very rapidly. As you can see in this study by Gervasi and Chisel, in which they prove that if you grow a population of brassica fast cycling in only presence of bumblebees. After only 11 generations, they will observe an increase of the attractiveness to bumblebees. In this paper by Roels and Kelly, they prove that if you grow Mimulus guttatus in absence of its pollinator, the monkey flower will start to reproduce by selfing after only 4 generations. It's also very fast. However, we have seen that most studies are interested by interaction between 1 or 2 pollinator species and 1 plant species. This interaction, called specialist interaction, is supposed to have different evolutions than generalist species. And today I will focus only on a generalist interaction between plant and its pollinator community. In a second time, we also know actually very little about adaptive response of plants in a natural condition. And so today I will focus on an experimental evolution that was performed outside in very changing environments. Together we will try to answer to those 2 questions. So the first one is how do phenotypic traits and fitness components correspond if we manipulate the pollinator access of the plant. And in a second time, we'll see together if we see any phenotypic changes, if these changes also change attractiveness to pollinator. To this end, I did an experimental evolution in a common garden. So for 6 generations, I grew 9 populations of plants. So each population was 36 Brassicarpa fast cycling. And each generation, I performed a pollination treatment on all of the populations. The 3 population treatments are those ones. So the first one, it's a control treatment. So for this one, I placed all the plants inside a cage, and I never opened the cage that was surrounded by a net, so no pollinator could come inside. And I did all the pollination by hand. The second treatment is called a full access. So it is a full access of the plants to the pollinator community. For this one, once again, the plants were inside the cage, and I opened the cage for 5 hours, so all the pollinator could come inside and pollinate. My last treatment was called limited access treatment. And so for this one, it's very similar to the full access, but I opened the cage for only 1 hour. So very few pollinator, and also very few pollinator species could come inside and pollinate. This experiment took place in the Botanical Garden of Zurich in Switzerland. And here you have a view of the inside and of the outside of one of the cages during its pollination treatment. And we could observe quite a large diversity of pollinators, including bees, bumblebees, and also butterflies. After this experimental evaluation, I wanted to compare the first and last generation, but because of different environmental conditions, it was difficult, so we had to do a resection experiment, during which we could reduce both maternal and environmental effects. During this resection experiment, I could measure morphological and phenological traits, elementary fitness components and compound fitness proxies, and also pollinator preference. In the first time, I wanted to test the mating system of the plants. So here I'm testing the outcrossing efficiency of the plants. So after hand pollination all the plants, I decided to measure the length of the fruits. And here what we observed is the decrease of the fruit length for the limited access treatment. Actually, when I was doing the pollination treatment, the hand pollination for this resection, I realized that the stamens were very reduced, so we had less pollen. And I think this could be the reason why we observed a decrease of the fruit length. In the second time, I also tested the selfing efficiency of the plants. So once again by hand pollination, but this time with its own pollen. And I measured the fruit length, and surprisingly, we found that the fruit length of the selfing fruit of the full access increased. So this result is a little bit surprising, and at this point, we are still not sure how to interpret it. So if you have any idea, feel free to come see me during the coffee break, and I will be very happy discuss about it with you. After this mating system, we wanted to see the evolution of flower morphological traits, as you can see here for flower diameter and ergogamy. And actually, we didn't find anything. There were absolutely no evolution of those traits. And actually, we think it's because we found very strong correlation between all the flower morphological traits and the climatic conditions. So for example, here with the main temperature and the main light. And during my experimental evolution, because we had one generation for every season, we had a lot of variations of the temperature and of the light. So we think that this variation of the climatic condition and this strong correlation could actually have prevented the selection on the flower morphology. However, we still observed some phenotypic evolution, including here the number of inflorescences. We've seen an increase of those inflorescences in the full access and limited access. And we've also seen an increase of some floral volatile organic compounds in the limited access and in the limited access treatments. And since we have this evolution of some morphological traits and phenotypic traits, we wanted to see if there is also an evolution of the attractiveness to pollinator. So to this end, I did a 4-choice test. So basically, in one cage, I put one plant from each of my 3 treatments and one plant from my initial population. And I released one among 2 pollinator species. Those 2 species were actually observed during my experimental evolution, so I know they were natural pollinator of my plants. And after releasing the insect, I was looking on which plants the insect would land first. Thanks to that, I was able to compare the different treatments and see which treatment was more attractive to my 2 pollinators. And my first result is this one. So we've seen that overflies, they have a strong preference for the initial population over the limited access. So there is a decrease of attractiveness if there is less pollinator. Actually, it is a decrease to overflies. On the other hand, when I was looking at the result for the bumblebees, I've seen that the control treatment was chosen more often than the full access. So there is also a decrease of the attractiveness for the full access treatments. And we think actually it could be due to the preference of the bumblebee for a higher number of flowers, as you can see here. And it's also something we found with the overflies. We also found some other traits that were influencing the attractiveness and the preference of the pollinators, including the ergogamy. We've seen that the bumblebees they prefer was the ergogamy, so when the pistil is smaller than the stamens. And we've also seen that medyl benzoate was not much preferred by both bumblebees and overflies. And to conclude, so we've seen that with my experiment, we could see changing in the mating system, so both for the crossing and the selfing efficiency of the plants. In the second time, we've also seen that there is a rapid response in floral scent composition and also in the number of inflorescences. But we didn't see a change in the flower morphology. And actually, we even see a small decrease of the attractiveness for the limited access treatments. And as a take-home message, I think it's very important to study plants and plant population outside in a natural condition, because we really don't find the same thing than in a very controlled greenhouse condition. And I think also it's important to both consider this kind of study on the short and on the long term. And on this, I would like to thank everyone here for listening to my talk, and also the organizer for organizing this symposium and also these sessions. And thank you. I think. No, you.
Okay. In your limited pollination treatment, the 1-hour treatment,
Mm-hmm.
did you see reduced fruit or seed set? Like, how effective was that treatment versus the
So for this one, I've seen a reduction of the overall number of seeds, and it was a very huge reduction. So I know that the selection on this treatment was very, very strong in comparison with the other ones. For the fruit lengths, I don't think I've seen anything actually. It was more the number of fruits in general. And also, not, so for the full access treatment, I've seen that most plants had fruits, but for the limited access treatment, actually very few could produce seeds for the next generations. And I think, yeah.
I had the exact same questions, but I can ask, did you take some measures of like overall fitness of plants, like at the end, like was the open pollination treatment resulted in like greater, I don't know, plant size or, you know, fruit set?
So for the plant size, no. Actually, I've seen that the control treatment has a small increase of the plant size, but it's more like the age of the plants. For the full access treatments, we've seen an increase of the number of branches, but I couldn't measure the overall rate of the plants, so I'm not sure if it's correlated to a reduction of the plant size or not. But it's something I would like also to test.
Hello, everyone. So I will present you in this talk some works that are part of my PhD. So I'm doing my PhD in Montpellier with Pierre-Olivier Chepteau. And I'm studying rapid evolution of selfing and of selfing syndrome. So this talk will really answer and call the previous one, so it's very funny. First, to begin, angiosperm sexual reproduction is the interaction between two sexual partners, like in every sexual reproduction, but for many angiosperm, it involves a third actor, which are pollinators. And I think that it is really, really interesting because it's not just a question of male and female and preferences between the two, but also preferences of pollinators that are carrying pollen. And as you can see, these pictures are carrying a lot of pollen sometimes. But sometimes some angiosperm are freeing themselves from pollinators by reproducing through self-fertilization. Evolution of selfing has occurred so many times in angiosperm that it is considered as the most common angiosperm transition, evolutionary transition, in angiosperms. If you look at this picture, you have each time flower on the left that is outcrossing, a species that is outcrossing, and on the right, a closely related species that is selfing. And if you compare these two species, you can see that the selfing species has smaller flower, less conspicuous, with male and female organs close together. And this set of traits presented in most of the selfing species has been called selfing syndrome. However, how this syndrome evolved and how it is correlated to selfing is not well understood. And it's a bit the topic of my PhD, and during which I used 4 populations located near Paris of field pensy, Viola arvensis. This population was sampled 2 times, the first time in 90s or 2000s, depending on population, and the second time in 2021. Then we used resurrection ecology methods, which basically consist in growing conserved ancestral seeds beside their descendant, which evolved in natural conditions. This allows to directly compare ancestors and descendants and to measure trait evolution. We choose Viola arvensis because it is an interesting model because it's an hermaphroditic annual with a mixed mating system, and it's a weed of culture. And in this culture, we will likely think that there is a decline in pollinators in this area. So, using population genetics, in the previous study, we we measured an increase in selfing rates within 30 years in the four population, and this increase is of 27% in average. So it's a huge increase in selfing, and we also measured several traits commonly associated to selfing syndrome, like floral color size, number of nectar guides, nectar volume, and so on. And here you have this trait represented in a multivariate analysis. And you can see that ancestral population, which are the triangles, are on one part of the graph, while descendant ones, which are dots, are on another part. And the left upper part of the graph corresponds to larger flora with more guide, more nectar. And so this demonstrates a rapid evolution within 30 years in natural condition of a selfing syndrome in our four populations. So this result was really striking, but we have an increase in selfing rate, an evolution of a selfing syndrome, and we wanted to know if there is a link between the two. Are the traits of selfing syndrome affecting the ability of selfing, or respectively ability of outcrossing? Because in Viola avensis, there is no pollen limitation. We didn't measure any pollen limitation, so the selfing events are directly limited by the number of outcrossing events. And a corollary question is how these traits are affecting the selfing ability. Is it more through male function, which means through siring out cross seeds, through exporting pollens, more or less, or through female function, which means through promoting ovules outcrossing, import pollen? To test it, we performed an experiment with a parental generation constituted of 8 populations of 32 individuals, which we genotyped and phenotyped. In each population, we introduced a bumblebee hive to open pollinate our individuals and collected seeds from 20 mother plants. This seed was used to grow an offspring generation composed of 5 offspring per mother plant, and this offspring was also genotyped. So we can assign them to inbreeding or outbreeding events, and if it's an outbreeding event, we can know who is the father. So if you take, for example, I don't know if you see this, probably. The blue pansy here, it has 2 outcrossing events through the female function and 1 through the male function. And the idea is to correlate traits to the number of outcrossing events through each function, male and female. The first result is that most of the traits constituting the selfing syndrome are well affecting the selfing ability under open pollination. But for this talk, I will only present you 3. Sorry. And so the first one is anthesis duration. You can see, on the right graph that anthesis duration is positively correlated to the number of outcrossing through the female function, while male outcrossing is not affected by anthesis duration. If we try to link it to the pollination, it's likely due to the short life expectancy of pollen measured in the Viola genus, which is lower than the minimum anthesis duration observed in Viola avensis. And the fact that anthesis duration is positively correlated to the number of outcrossings through the female function implies that late outcrossing can occur in Viola, which corresponds to a wait-and-see strategy regarding selfing, and which is an interesting thing to know to well understand how it happens. To conclude for this trait, so you have a positive effect of female function on the outcrossing events, of anthesis duration on the outcrossing events, sorry. For among tested traits, few are affecting outcrossings through both male and female functions. The number of nectar guide is one of these, and it shows a positive effect on outcrossings through male, on the left graph, and through female function. So this trait has concurring effect of male and female function on selfing also. But if we look at another trait which affects both function, which is mean number of aperture is it's a characteristic of violavensis which present pollen heteromorphisms. Pollen heteromorphism is the fact that a single individual can produce several pollen morphs. So as you can see in the picture, some pollen grains are 4 aperture rates, some are 5, and some are 6. These 3 morphs are found in a single individual, in a single anther. And the proportion of morphs differ between individuals, and the differences can be summarized calculating the individual mean number of aperture. And so if you look at this mean number of aperture, you can see that it is positively correlated to the number of outcrossed offspring to the male function, but negatively correlated to the number of offspring to the female function. And this is due to, a number of pollen aperture is positively correlated to the speed of germination of the pollen tube. So it implies gametophytic competition between the morphs. So plants producing pollen with many aperture have pollen grains germinating faster than others when they are deposited on a stigma, and therefore outcompete other pollen grains. So outcompete other males is interesting to outcross ovules of other plants, but it limits the ability of other males to outcompete its own pollen deposited on its own stigma and to outcross its own ovules. So our result demonstrates the opposite effect of these traits on selfing through male and female function. And what is interesting, we can associate this function of traits to the evolution rate of these traits during evolution towards selfing. If we look at the anthesis duration, which is, I remember, positively correlated to the number of outcrossing events, it's well counter-selected during evolution towards selfing. But, and it's also the case for nectar guides. But you can see that the evolution rate is higher for nectar guides than anthesis duration. And it's not due to a difference in heritability. We measured the heritability, and heritability of nectar guides is even lower than the one of anthesis duration. It's likely due to the fact that nectar guides are selected through both sexual function, male and female functions. And finally, if we look at the evolution of mean number of aperture, it doesn't evolve rapidly. And another time, you have a high heritability of this trait, so it could evolve. Its stasis is probably due to the fact that male and female function are affecting outcrossing in similar magnitudes, but in opposite way. So to summarize, traits constituting the selfing syndrome are well affecting selfing. Some traits are affecting selfing through male or female function, and some through both in concurring or opposite ways. How traits are affecting selfing is an important factor to understand the evolution of selfing syndrome, and moreover, the space of this evolution. What is also really interesting is to link these traits to pollinator behaviors. And I had some data about it, but I haven't the time to present it. So if you want to discuss it, it will be really interesting. And moreover, another thing that is really interesting about the function of traits, it is that it depends on the context. Some traits are blind to the other mate opportunities, and some not. So if you want to discuss it, come to talk with me. Thank you very much for your attention, and I'm pleased to answer your questions. No question.
So do you know anything about the relative fitness of self versus outcross seeds in the system and the environment? Like, is there evidence for the strength of inbreeding depression and how that might play in?
We measured inbreeding depression, and we find almost no inbreeding depression in this species. This species has already a selfing rate of around 50% in the 90s, so we can imagine that it already have been purged. What?
So in the resurrection experiment, Do you have an idea of how much those populations were isolated? Like, were they changing genes or?
Yeah, we consider our whole population totally isolated because we measured FST, and the spatial FST was really high compared to the temporal one. So yeah, it's basically 4 parallel evolution of the selfing syndrome. And even which is interesting is that it's not always the same traits that evolves. So perhaps some differences in pollinator communities or pollinator declines dynamic. Yeah.
Super interesting work, thank you. How do you or plan to measure the fitness of the sexually and asexually used offspring?
It's what they almost have the same fitness. We try to measure it, and we don't find any differences of fitness. We don't find any inbreeding depression.
I think we're out of time already.
Yeah.
Maybe you guys.
Thank you.